Hi everyone. So this is from Vivid Botany. We have a question here that is from biotechnology principles and process. During the purification process for recombinant DNA technology, addition of chilled ethanol precipitates out. See, the question is extracted from biotechnology principles of subtopic isolation of the genetic material. See, first, to obtain the desired DNA, we have to isolate the genetic material from the cell. So, the process is here isolation of genetic material. What acts as genetic material? We know it very well. That is DNA, dsDNA acts as the genetic material. So, this genetic material, which we call it as the DNA, present inside the cell. So, first we need to break down the cell wall. That is, I write it here, digestion of cell wall. If the cell wall is of plant, then we apply here the enzyme cellulase. It can be pectinase or it can be hemicellulase, like that, because we know that plant cell wall is made up of cellulase, hemicellulose, pectin, polysaccharides, etc. If it is fungal cell wall, the cell wall is digested by the enzyme called as chitin. Chitin is a complex polysaccharide homopolymer of polysaccharides present in the cell wall of fungi, and chitin is second most abundant organic material present on this planet. Chitin is present in also the cockroach, that is Periplaneta americana, in which the outer shell is made. And if the organism is bacteria, then the bacterial cell wall is broken down an enzyme called as lysozyme. It's an enzyme which will digest the bacterial cell wall. After digesting the cell wall, we get the plasma membrane along with protoplasm. So, the protoplasm is subjected to undergo centrifugation to isolate the different cell organelles, that is chloroplast, mitochondria, ribosomes, endoplasmic reticulum, Golgi complex, lysosomes, peroxisomes. They isolate according to their densities by the process centrifugation. Thereafter, isolation of all the cell organelles along with the genetic material, we know that the RNA proteins are present. So, proteins are digested by an enzyme called as protease. Proteins are digested by an enzyme called as protease. And thereafter digesting the proteins, we get the RNA. That RNA also associated with the DNAs. So RNA get digested by ribonuclease. Ribonuclease can be also called as RNase. So by which RNA gets digested, and at last we get the DNA. After isolation of all the components, we get the DNA. So this DNA is precipitated in purified form by addition of chilled ethanol. Purified DNA can be precipitated by addition of chilled ethanol. Ethanol, we know that it is ethyl alcohol. We have a mention of ethanol in various forms like that. I remember from yeast, ethanol is obtained, and when ethanol concentration increases more than 13 percentage, that causes death of the yeast in fermentation. And ethanol is a product of anaerobic respiration. It causes intoxication, inebriation, and it is the main product of toddy, that is South Indian traditional drink. So while making molasses, from molasses also ethanol is prepared. So in this question here, during the purification process of recombinant DNA technology, addition of chilled ethanol precipitates out. See, purified DNA is precipitated by adding chilled ethanol, that is DNA. And it's not histone proteins, it's not the polysaccharides. Polysaccharides are nothing but the carbohydrates. Histone proteins are basic proteins that are associated with DNA. We see that H2A, H2B, H3, H4, H1. So, four types of histone proteins are present in nucleosomes. They are called as intranucleosomal proteins. H2A of 2 molecules, H2B 2 molecules, H3 2 molecules, H4 2 molecules. Total 8 histone molecules will make up one nucleosome, and H1 histone protein will be present outside the nucleosome, that is locking histone protein, that is locked, unlocked during the condensation, decondensation of the chromosome material. And histone proteins are made up of basic amino acids like lysine and arginine. So histone proteins associate with the DNA. We know that DNA is acidic and DNA is negative, and DNA migrates during the electrophoresis technique to positive electrode, that is anode, because of its negative charge. And this negative charge DNA gets neutralized, compensated by addition of basic histone proteins. That electric stability of DNA is increased by association of the histone proteins. So that is about histone proteins, and here the right answer is DNA. It is one of the process of recombinant DNA technology, chapter biotechnology principles and process. The question is very simple. One should go through all the processes very clearly. So I think my explanation for this question is very much useful to you. So do see the remaining questions also one by one, and try to concretize your concepts all this from this chapter, and try to subscribe my channel also. I hope my videos will be better useful for you. Thank you.